Okay. Thanks everyone for coming. And I'm just going to quickly introduce Michael. I'm filling in for Ian, who is unfortunately sick right now. But Michael and I go way back. We were actually friends since freshman year of college. And yeah, we even took Ian Cousin's animal behavior course together. Well, somewhat. You showed up. But Michael has always been very interested in bees, and yeah, he even started the beekeeping club at Princeton University. And he's then went on to do his PhD work at Cornell with Tom Seeley, doing some really cool work on sort of understanding how honeybee hives detect their colony size, right? He then came here to do a postdoc with Ian, doing some what I hear is extremely daunting work, just tagging thousands of bees and observing them in these observation hives. And currently he's, a professor now at Auburn University, and has a fantastic lab looking at honeybee nest construction. And he's also currently at the Vico in Berlin for, I guess, a sabbatical. What do you call it? Fellowship? Who knows? Having fun. But with, no further ado, Michael Smith will be talking about nest design, construction, and spatial organization in the superorganism, which I think are honeybees. Okay.
Cool, perfect, thank you. Thank you everyone for being here. So my name is Michael. Thank you, Edward, for the introduction. I'm sorry Ian couldn't be here. This was like a huge part was to see Ian, and it's great to see so many people that I know, but also a ton of people I do not. But this actual talk title should probably be, Yo, Michael, what you been up to? And maybe I need some bees, and by I mean you. So if you're in this talk and you're like, wow, like maybe I need some bees, maybe I would be the person you would want to bees with. Because you'll notice that I am at Auburn, but I'm also an affiliate at the Max Planck Institute. So I'm really, really excited to work with people at Uni Constance. So kind of consider this as a pitch. But this talk is going to be all the work that I've done since being at Constance. So actually, unfortunately, I'm not going to talk about anything I did while I was here with Ian's group, which I loved working. I love working with the people here at Constance. So think of this as almost as like a pitch for the future. Edward's totally right in talking about what my lab does. We're really interested in honeybees as an integrated superorganism, and the lab's work is really integrating animal behavior and evolution using novel tools to understand how these superorganisms survive, grow, and reproduce in a natural history setting. The three main things that you need to know just to kind of get a baseline from going onward so that some things make some sense is there's three concepts. One being the superorganism concept, which I mentioned in the title, but I didn't necessarily explain. The second one being the extended phenotype. And the third one is a realization that evolution is acting on developmental systems and that it's the entire developmental organism that's important. So because we're all biologists here, or at least biology adjacent, I think we'll probably be able to go through these kind of preemptive precursors pretty quickly. The first one being the superorganism as a unique level of biological organization. And so if you think of like a unicellular organism, you can think of many unicellular organisms coming together and forming a multicellular organism. But in the same way that you can think of that kind of transition, you can also think of many, many multicellular organisms coming together and forming a superorganism. So these are traditionally thought of as the ants, bees, and wasps. Obviously, the honeybees would be my preferred superorganisms, but people working on ants, termites, wasps, these are all these kinds of classic superorganisms. And that analogy has been applied in a lot of different contexts, but in these cases, it's that it's a unit of selection, and this is an integrated organism that evolution has shaped. The second key concept is the idea of the extended phenotype, and that evolution acts beyond our actual bodies and the things that we actually build that can also help us survive, grow, and reproduce. And I'm putting up a lot of different beautiful examples of extended phenotypes, whether that's a beaver's dam or a spider's nest or a bird's nest or a termite's nest or a honeybee nest, all these things that organisms build that help them acquire resources, attract mates, communicate, anything that can help them do the things that they need to do can be part of their extended phenotype. And that extended phenotype itself is under selection. The way a bird builds its nest, there's a reason behind that building materials that they use and the structure that they build. The last key concept is the idea that organisms are not just adults, they are life cycles. So obviously I'm going to use this as an example to kind of get you up to speed on the honeybee life cycle. A honeybee colony begins as a swarm with a single queen and about 10,000 bees. They've clustered on a bush here, in this case a nice beautiful rose bush with the lab truck in the background. And that swarm of bees is going to find a new nest cavity, so in this case a tree with an empty cavity inside, and then they will move into that cavity and build that nest, stockpile that nest, and then hopefully, if they survive their first winter, they will then produce drone reproductives and additional swarms. Now, this is an entire life cycle of this organism, and evolution can act and does act across that entire cycle. What types of cavities are considered acceptable? Evolution can act on that. How, when, and where to stockpile your nest. When to make these kinds of divisions. These are all things that are part of the entire life cycle of what it means to be a honeybee superorganism. So with this, I'm going to use a couple examples to kind of combine these concepts to kind of give you an idea of why I think that my lab at least is working at kind of an intersection between these areas that potentially is interesting. So the first thing is that the nest architecture itself also has a life cycle. In this case, what we're showing is a single comb of the nest and how it grows over time, with the color of the lines showing you the progression over time as it's built. And this actually also exists in three-dimensional space. So this is a case where we've actually taken nests and installed them into combs, and then we can extract the outlines of those combs, and we can show that a honeybee colony doesn't just jump into a nest and just build all willy-nilly. They actually build a nest in this kind of a three-dimensional stereotypical fashion. Similar to the ideas of your human body or a unicellular organism being spatially organized, the nests are also spatially organized. And this spatial organization is actually maintained over 34 million years of Apis evolution. Whether you're a dwarf honeybee like Apis florea, a giant honeybee like Apis dorsata, or a cavity nesting bee like Apis mellifera, you have certain regions of your nest that are devoted for different nest contents. And that's presumably for an adaptive benefit, but also open to interpretation and experimental manipulation. And so then to combine all those things, these nests being spatially organized, they also have a developmental trajectory. What a nest looks like in its first week of life is certainly not what that nest is going to look like in its 20th week of life. In this case, what we're doing is we're doing a rotating flyby. Each of these is an individual comb where the contents of each pixel cell are what that hexagonal cell contains. And I'll get into that later, how we actually extract those kinds of data. But this is all kind of coming to the idea of that spatial organization and the superorganism concept is a reality in the honeybee. And it's also something that we can truly manipulate and impact. So at this point, you may be thinking to yourself, well, Michael made a horrible mistake because Michael works with honeybees, and honeybees are the most well-studied social insect in the world, and he has made a horrible life, career, bad accident mistake. And I would say, originally, I probably would have agreed with you, because from a young age, I thought, well, honeybees, everyone knows everything about honeybees, so why would anyone work with honeybees? Surely this has been done before, and Michael, you're a nincompoop. And I would agree with you, but I'm going to show you, hopefully, or potentially convince you that there are some major mistakes that have been made in the field that maybe, hopefully, we're trying to start to fix. And I'm going to use yeast as an example of why we have huge gaps in our understanding of honeybee biology. So if you think of yeast and honeybees, these are two organisms that we have been interacting with for thousands of years, and it's because they have things that we want. And in the case of yeast, we use yeast to do things like bake bread and things like brew beer and wine, and these are products that we want from the yeast. But in order to get those products out of the yeast, we need to understand the entire life cycle of the yeast. So for example, what kills yeast, what helps it thrive, how to actually control these kind of different processes. The difference, though, with honeybees is that we have similar things. We have products that we want for them. Historically, we wanted honey from them, and we wanted wax from them. But because of the way that we keep bees, our modern beekeeping methods, they're actually 100% divorced from the natural life cycle of bees. And that is namely the movable frames that we use to maintain bees makes it extremely easy to manage honeybees, but also means that we never have to actually understand their development to actually get more bees or to get the things that we want from bees. So here's a classic example. This is the Beekeeper's Manual, most complete and essential list of your beekeeping needs. And it is 1, 2, 3, 4, 5 boxes deep. A natural honeybee colony would only live in a single box. Versus this is like your kind of standard thing. And these boxes allow you to mix and match and divide and subtract. You can call up someone and say, hey, send me 10 colonies, and that's all fine and good. And really, a lot of people never have had to actually kind of carefully document a lot of the developmental parts of colony life history. And I think the most embarrassing example of that for me personally was that during my PhD, I was doing a sociometric study of colony development where I was literally just taking plastic sheets and tracing things out. I'll show you this later. And it was only at the end that I realized that no one had done that before. So there are some really serious gaps in honeybee biology. I'm not saying that bees are clearly very well known in some ways, but there are also some exciting gaps in there too. So if I were to give a tagline of what the lab actually does, I'd say we're all about how and why honeybee colonies do what they do. And that's because our levels of analysis are both ultimate and proximate. We're looking at the colony level. Those are our level of biological organization. And do what they do means that we're really trying to link up to their natural history and ecology. We're not necessarily interested in how this connects to a beekeeping context, but more the basic biology of these things. So with that, we're going to go through two parts, two like kind of vignettes in this, and hopefully I'll convince you of the importance and the value of working with honeybees and really relying on your eyes to see things that the bees might be showing you. So the first part is the evolution of cell building behavior. And we got your classic beautiful photo of bees and comb building. And there are many people that have thought about and looked at comb building and been amazed by it, including Darwin, the most wonderful of all instincts, or Pappus of Alexandria, thinking that they had some geometrical foresight. But the kind of big flaw in there is that natural nests are actually not perfect. Everyone historically has been obsessed with the perfection of the hexagon, but when you actually look at what bees do, they don't actually build perfect nests. And their nests are not perfect for two reasons. Well, I don't want to say they're imperfect, but there are two things that bees have to overcome. The first is that they build two different sizes of hexagons in a single sheet of comb. They build small worker cells for building worker bees, and they build larger hexagons for drones, the male reproductives. But that's two different sizes of hexagons in a single lattice. So that is an architectural challenge for them. The second problem that bees have is that unlike the beekeeping equipment that we keep them in, they're often building in irregular cavities that have sticks, that have curves, that have all kinds of different oddities in their nest, and they have to adapt their nest to that shape. The third thing that bees have that can cause architectural problems is that they often build comb in multiple places at once, and then they have to put those things together. So in this case, you have two independent combs, and the bees are going to merge those together, and they have to actually fix those architectural challenges on the fly as they're going to go. So these are lots of different reasons why their nests are not perfect, but typically, as researchers, we just ignore those imperfections. And you might be wondering, well, why would we ignore those imperfections? And the simple answer is that they're really difficult to study. It's hard to study all the non-hexagonal, all the weird things that bees build because they just don't kind of fit the standard, okay, it's a ton of hexagons and how perfect are they? My favorite example of this is actually Heinrich, who measured over 4,000 measurements of hexagonal cells, but didn't do a single measurement of a non-hexagonal or an irregular cell because he's like, I can't touch those with a 10-foot pole. Hepburn and Whiffler in the 90s, they would give combs a pass-fail based on whether there were any non-hexagons or not. So, maybe for them that was fine. For me, that seems a little harsh. And then here we have a researcher very close to my heart that started doing this by hand and then quickly realized that there was no way he was going to finish this project. And that was me, and this is my notebook from when I was a graduate student. And you really quickly realize with this set of calipers that you're really, really in for a lot of misery if you try to do this by hand. So what this meant, though, is we needed some kinds of new tools to explore these old questions. And so for this, I collaborated with Kirsten Peterson at Cornell University. And I realized there are different ways that one can do this now. But at the time, we basically used some simple methods of image analysis to basically automatically first get blob detection for the center of the cells, link, and then use basically pixel. You're basically walking along the line to find the point of greatest pixel difference. And then you basically draw a perpendicular to get your vertices, and you get a hexagonal outline of your cell. So I'm obviously showing you a toy example of this. I'll show you some other examples after, but it moves you a lot faster. So whereas you'd have to do this by individual cells, we can actually go through an entire comb with multiple thousands of cells in about 15 minutes. We can actually go through and correct that entire thing. And that gives us the individual metrics for every single cell, cell walls, side lengths, angles between them, and everything. The experimental methods we're actually using, though, are very, very simple. Our plan of attack is basically, hey, bees, build some comb, and then we're going to look at it. And that's about as simple as it gets. So this is my hand on the left-hand side. You can see that very nicely these gals have started building in 5 locations, and then they're going to have to now, they've now created naturally these problems that they're going to have to overcome. I come back a little later. They've kept building. Nicely done, ladies, but not done yet. And then they've now filled out this comb, and then we can take it into the lab and then get a nice high-quality image. And this single frame can showcase a lot of different challenges. If you've got a good eye, you might even be able to, you can maybe even remember where those different pieces lie and where those kind of like different irregularities were. So this is the example image that I'll use in a lot of these cases, but just know that the data set actually includes a lot more combs from a lot of different colonies. So how do bees fix these architectural problems? And I'm only going to focus on 2 specific problems. There's many more that we can talk about if you'd like to afterwards, but these are like the kind of key ones that I wanted to compare and contrast. The first problem is, how do you transition from small worker cells to large drone cells? And the second one is, how do you merge 2 pieces of comb, in this case, a worker comb with a piece of drone comb? So it could be worker-worker, it could be drone-drone, but in this case, it's worker-drone because you basically have worker to drone or worker to drone having to fit them together. So let's just measure the cells and see what the bees did. So in these plots, they're all going to be oriented based on the cell position of when you swap from worker to drone. And then these shading areas are going to give you the 95% confidence intervals for what is considered a worker cell versus a drone cell, given no problems for the bees. So if you just give the bees a total piece of comb without any kind of issues, this is kind of the regularity that they would build their cells. And so this is over 2,000 cells are in this plot. Each individual dot is an individual cell. And what we're doing here is we're basically walking along a line where they're building worker cells, worker cells, worker cells, worker cells, and they go through this very slight transition in cell area where they get to drone cells. All the cells that are, all the data points that are orange here are all your 6-sided cells. You see a couple that are different colors. That means that they have different numbers of walls. So they're these non-hexagonal cells. But for the most part, they're just going through this really smooth transition from one to the other. So let's contrast that with what happens. So those are the intermediate hexagons. Let's contrast that when you actually merge from 2 sides. So this is where the bees are building from here, and they're kind of moving inward, and they're going to now have to kind of fix in the middle. And you'll see some things that are similar. So we still see this kind of general transition between the 2, but there's a lot more noise around that area, a lot more of these other colors that you're seeing. So a lot more of these 5-sided cells, 7s, occasionally an 8 in there, 4-sided cells. They're building a lot more of these irregularities in there. And what this was telling us was that it's a lot harder for bees to merge independent pieces of comb together than it is to go through that really gradual transition. That'll come in mind in a second as well. So what makes merging so much harder than transitions? And part of why this was something that I was interested in was I spent so much time just looking at these images and just seeing what the bees had done. And this is an example of there's 2 pieces of comb here that the bees have merged together, and it looks like flawless. I mean, it's really a beautiful instance of collective construction here. And if you've got a critical eye, you'll probably notice that there is a single non-hexagonal couple in here, which for me, I'm like Hepburn and Whiffler. Like, really, you call this a failure? They had one little mistake in there and the gals are in the trash? That's no good. But because we had this new method, we could actually quantify the tilt in every single cell, because that's kind of like the idea we're getting at. If you think of the pointy bit of the hexagon versus the flat part of the hexagon, when you have those two things coming together, that's where it's really going to be hard to kind of overcome those challenges. So when we come back to that original image I showed you, we can actually color every single cell according to its tilt. And then we can quantify the difference in those two tilts to give us an idea of how more difficult or less difficult a problem might be. Because at the end of the day, it doesn't matter what your tilt is, it matters how different one is from the other. And sure enough, what makes merging harder than a transition is as the tilt difference gets bigger, you have more and more non-hexagonals built. So this is here the cell position and the proportion of non-hexagonal cells. As you go from a first quartile, second quartile, third or fourth quartile difference in the two sides, you actually get a higher and higher proportion of non-hexagonal cells at that merge point. All that is to say is that it's really hard to fit hexagons together when they're not aligned. And they can't coordinate that alignment if they're building independently in those two different places. But because we could actually look back at these images and because we had this new tool, we could also look at, well, what's the actual pattern of all those different non-hexagonal cells? And if you've got a good eye, you might notice, so first off, orange, I'm using the same color. By the way, if you're colorblind, these should be colorblind safe. But occasionally I've talked to people who are colorblind and be like, Michael, that you can't get 5 colors and still be colorblind safe. So hopefully this is kosher. But so all these orange dots are for all of our hexagonal size cells, and I made the circles bigger for all the non-hexagonal cells. And just being able to look at these, you might actually be able to pick out some patterns in these non-hexagonal combinations. Namely, yellow and pink, 5s and 7s are often right next to each other. Also, 7, 7, 4s, these little couplets, they're these different motifs. And what this was telling us was that the bees aren't just willy-nilly incorporating all these non-hexagonal cells. There's actually some kind of predictability in what type of non-hexagonal cells get incorporated. And that's going to become important in a second when I go wayward and I kind of abandon my true love and I kind of get a little bit, I dabble in some other social insects. Because Apis mellifera isn't the only hexagon building collective. And Edward is totally correct. I love honeybees. I love working with honeybees. But occasionally, I get a little restless and I feel like I need to see what the other bees and wasps are doing. Occasionally wasps, mostly just other bees. And so what this meant was, well, we can see how honeybees can come up with these solutions to architectural problems, but how are those solutions shared or different from other species of honeybees and other social wasps? So for this, we needed to do a collaborative work, but there were reasons of why you might think that they would be similar or different. So here's an example of honeybees building their comb. They build it this way. They build their comb out of wax. Their comb is double-sided. The wasps, in contrast, they build their combs facing downward, single-sided, out of paper. So really what you have is you have different materials, different configurations, also different evolutionary origins. Bees and wasps independently arose at the hexagon. So it's not a shared origin. But the same is that they have the same architectural problem. And I'll get to what that architectural problem is in a second. But what was useful about this is social insect researchers are also very social, and you could kind of call out to a lot of different people, and through their networks as well, get a lot of images. So I have to give a shout out to Bajiri and Tatsuya, Yana, James, and Kevin, who through them and their networks and many other people, we were able to amass a very large data set of images. The fortunate thing is that we got a lot of images. The unfortunate thing is that none of them were as good as the honeybee images that we got in the lab. They were like these beautiful, side-lit, easy to annotate things. They were like messy trash. But fortunately, I just had a kid at the time, so I had no brain power, but I could click on a lot of images. And so that's what I spent my parental leave doing while like rocking a kid with one foot. But with this, we were able to get 5 species of Apis, mellifera, florea, Andreniformis dorsata, and Apis cerana, and then also 5 species of social wasps. And here's a phylogeny of those different species. So they're independent evolutions of hexagonal cells. I use brown to indicate paper for wasps versus yellow for wax to indicate the honeybees. Originally, I had illustrations of bees and wasps, but I realized that probably no one would tell the difference. That's fine. And the shared challenge that they're all going to have is for many of these species, the reproductives are larger than the workers, and therefore they're going to have to incorporate those different size hexagons into the same lattice. So that's the similar problem, even though there are different groups. And the scale of that problem is going to differ on the species based on how much bigger their reproductive is than their worker. So in this case, we have a control that has no size dimorphism. These ones have very small dimorphism. Apis mellifera, unfortunately, was our lowest size dimorphism. So this is where the workers are, this is the drones. And then basically we've organized them going onward in terms of larger and larger dimorphism. So as you move along that scale, you're getting larger and larger differences between the size of a worker cell and the size of a reproductive cell. So how do all these other collectives fit hexagons together? For these species that don't have dimorphism, not surprisingly, they don't have to do anything different. But then for Apis mellifera and all these other species, we're basically finding all of these similar kind of transitions, and then also the pink and yellow, the 5- and 7-sided cells that are happening at that transition line, that point at which they're switching from one construction state to a different one. And what was even more convincing for us was to show that because we could actually go back to those images of the comb and actually orient them based on the order of the cells that were built, we actually find that in every single species found, if you look at the direction of comb build, they're always putting a 5-sided cell and then a 7-sided cell. No matter what species we look at, it's always a 5 and then a 7, 5 and a 7. They're always contracting one cell and then expanding the next one to kind of accommodate those two different sizes. So what that's telling us is that they're all using the same architectural tricks. Regardless of species, they're using intermediate sized hexagons and these 5-7 pairs. And so what we thought at least is like, well, this is pretty cool. This is some kind of architectural convergence in terms of these nest building problems. But could we kind of model this in a way that would incorporate those oddities instead of avoiding it? Because typically it's, in the past, it's always been like, oh, irregulars, let's just not look at those. So for that, worked with Niels. He's a brilliant mathematical modeler. And we basically just looked at a lot of different ways of how these bees and wasps could be doing this. And the first model is this multi-layer transition region, where you basically say, okay, I need to go from A to B. I'm going to just do it very, 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 very slowly in incremental steps, and you'll never incur a non-hexagonal cell. And so something like Vespula shadai here does something similar to that. Versus other species like Apis florea, they basically incur these 5-7s because they don't go through a very gradual transition. And we can actually predict where those, or the rate of those transitions based on the difference in dimorphism. So another way to think of this is for something like Apis florea, where you have very, very small worker cells and very, very large cells, if you were to go slowly over every single small slight addition, you would end up with a very large area of intermediate cells that they basically have already told us are not usable because that doesn't fit with their natural history. So this is the idea of comparative nest architecture, and that we can actually use these new tools to kind of investigate old questions and see how reproduction actually impacts their architecture. So to kind of span this out a little beyond the social insects, so here's what the social insects have done. They do 5-7 pairs when they have to combine these hexagons together. And well, the diatoms actually do the same thing. They also have 5s and 7s also in their structures. And even something without a brain, 100% graphene, graphene does the same thing. Graphene also has these 5-7 pairs when you actually create these grain boundary edges. So you might think that I'm convinced that the bees are geniuses, and I would maybe say that they are, but I would also say that graphene is definitely not a genius, and it kind of comes up with some of these similar. I'm sorry, I know, but graphene is not a genius. But these similar solutions are being found in other arenas. So at this point, I'd like to zoom out a little bit. From the cell level, we've been looking at just that individual cell and what the bees are building, to actually looking at the entire nest and what they're doing with their entire nest. And this is the part 2, the form, function, and evolutionary origins of this architectural symmetry. And I'll explain, hopefully, what I mean by that architectural symmetry, which is something a little strange. The first thing, though, is to give you a primer, is that in biology, at least, we live in a very symmetrical world. The vast majority of organisms that you ever would interact with are symmetrical. Our bodies are symmetrical, at least on the outside. We build symmetrical cars, planes, trains. We often like building symmetrical buildings, even though I know in the inside the Taj Mahal is not symmetrical, but that's a whole other thing. But that symmetry also doesn't necessarily span levels of biological organization. So if you open up a human body, the human body internal organization is not symmetrical. But the reality is that this project actually started with an unexpected observation. And this is coming back to actually work done over 10 years ago during my graduate school. And these were the observation hives that I was using to kind of extract these nest developmental patterns. So to do that, I set these colonies up in these large observations. So these are about 1 meter by 1 meter. And you basically dump a colony of bees in there, and then we just let them do their thing. And because we can see inside the glass, we can look at these colonies and undisturbed and say, okay, well, this is honey, and we have some cap brood or some young brood. There is an egg here, I promise you, or some pollen or some nectar. And you go basically through and you make a map of what that nest looks like. And that's what one side looks like. But colonies are double-sided. They have 2 sides of comb. And this is what the other side looked like. Which to me was surprising because it sure looked like a mirror image. And yeah, at least in the lab, we call this butterfly format, where we take a colony and we butterfly it. So you can actually see both sides of the comb at the same time. But this also caused a problem for me because I then realized, now that I've seen this magic trick, I can't necessarily collect this data myself anymore because I could potentially bias myself. So we had 2 exceptional undergrads that joined the lab. Claire Bailey, who's since gone on to do a PhD. She's not completed her PhD, but she's doing her PhD in New Jersey Institute of Technology. And Maritza went into data science. And she was very happy to do that after she looked up my salary online. So I told these 2 students, I said, look, here's the deal. We're going to do this project together if you're up for it. But I'm going to tell you beforehand that there are things I can't tell you about it, and you'll only be able to tell you at the end. And so I was like, okay, here's how you map these nests. And the deal is to share the work. Whenever you map a nest, you do one side, you do the other side. And so we're dividing the work. And I didn't tell them about the symmetry, and it was kind of like that was the way we did it. So here's what they came up with. This is a very small subset of the data of colonies that we then mapped in Auburn. And what you hope, so this is different people are mapping each side, and all you really need to know is that different colors are the same contents. And what this found was that for every single colony we've done, at every time that we've ever measured them, they're always symmetrical. This front-back symmetry is persistent, and it's both colony and developmental specific. So even when a colony swarms, even when a colony is on its deathbed, they're still symmetrical with their backside. And if you flip or swap one map from within the same colony or a different colony, they're no longer symmetrical. So this led to an obvious question for us, at least, was, do you need direct contact to create this front-back symmetry? So this comb is double-sided. You can imagine a scenario where, okay, I'm a bee, I collect pollen, and I come back and I put some pollen right here. And then when I come back the second time, I'm going to go back, I'm going to put it over here. Or I'm a nectar worker, and I'm going to work some nectar here, then I'm going to work it over here. So I want to know, do you need to have direct contact with both sides to create that symmetry? So for that, though, we wanted to build this very strange observation hive that we can manipulate this kind of system. And I call this the worst beekeeper ever because this is really stuff that we're using tools from beekeeping in a way that beekeepers would never do. And honestly, if you tell this to beekeepers, they often are like, why are you doing this? You guys are horrible people. And what we're doing with this is we're basically creating a single substrate that a colony can live on, but we can control whether they can pass to the other side or not. So we can install a single colony here, and then we can install a totally independent colony on the opposite side. So this is these plastic foundation, so it's impermeable for the bees. And then we mount it in the wood, and we coat it with a thin layer of beeswax. And then we basically dump in your bees. Don't ask how we get them in there. And then we basically let them build their nest over about, in this case, about 23 days. And then we come in and we map the nest. And then this is what that nest looks like. And just for a key so you know, I typically put brood in blue because it starts with the letter B, pollen in orange, and then light green and yellow as versions of nectar and honey. So this is what side 2 of this observation hive looked like. And this is what side 1 looked like of that observation hive. And the question for you is, are these the same colony or are these different colonies? And just as a clarification, when we put this same colony, we just cut little tiny corners out so that the bees can actually move from both sides. So do you think it's the same colony or? I feel like I've kind of primed you on it. You'd say different? Okay, so these are 2 independent colonies. Independent colonies, no contact with each other. We actually do cross-fostering things where we actually take bees from each side and swap them over to make sure that they're independent, and they kill them all. So like, we only do it with a couple, Giovanni, it's fine. It's not too many. But they're independent colonies, independent entities, and they're somehow mimicking each other's nest organization. We find that the symmetry is the highest with your backside counterpart, regardless if it's the same colony or not. So either you have single 8,000 worker colonies that are living on both sides, or 2 paired 4,000 worker colonies, each living independently. And we find the same level of symmetry, whether or not you're the same colony or not. So that led us to think, well, how are these independent colonies coordinating their nest organization? What kind of information are they kind of passing between the 2 sides? And for honeybees, temperature and thermoregulation are extremely important for colony function. So it was like an obvious cue for us to test. And also because we have this impermeable plastic layer in there, chemical cues didn't seem like a very viable thing to get through 2 millimeters of plastic or around the edge all the way back and then to tell a bee like, hey, put brood right here. So we went super high tech on this. We went to like a pet store, and we got these reptile pads, and we basically created a hotspot where we could randomly position this heat pad set to brood temperature wherever we want there, and then we install a colony on the opposite side. And just in case you're thinking, we always do all of this stuff blind. So anytime we come into the container to actually map these nests, you don't know where the heat pad is going to be. You're just going to go in, you're going to map a nest, and then afterwards it'll be, okay, and then here was where the heat pad was. So I'll give you a second to look at, so remember, blue is brood, and for the beekeepers, pupae, so the cap brood, the dark brood, is the oldest brood in these colonies. So tell me if you can think about where there might be a rectangular heat pad in each of these 6 observation hives. And so it turns out that's where the heat pads were. And if you're a statistician, have I got some stats for you? 100% of their cap brood was co-localized with the heat pad. So we definitely did a statistical test right there and gave it a star. So what this is telling us is that the temperature, what we know is important for bees, is a way for these bees to actually indicate where they're going to position their nest. Remember, they're getting installed in this box. They have no prior nest. They can literally put their nest wherever they want to in that two-dimensional space. So what this is telling us is that this temperature was somehow important for nest organization, kind of similar to like a Schwebon-Malengond organizer in developmental biology. The idea being that you have some point of initiation, and that's going to then induce other parts of the nest, and you create this whole kind of pattern. So we want to know, though, was there a benefit of living in this 2-sided nest? So here was our 1-sided nest. So we basically take that nest, and we can just have a colony live on one side of it. Like think of this as like pages of paper. We're just going to have you live on one side of that page. Or we're going to fold it on itself. We're going to give you 2, but we're going to give you access to both sides. So do colonies do better if they live in a 1-sided nest or a two-sided nest that has double-sided that can be stockpiled symmetrically. And what we find is that, yes, living in a two-sided nest lets you rear more brood, so they rear more brood, and they have less blank space than their one-sided counterpart. There's no difference in terms of how much nectar and honey they're able to produce, or how much pollen, or how much comb. That's not significant. And then the other part that was interesting was that the actual, we embedded thermocouples in these colonies, and while their mean temperature is the same, there's a lot more variation in the colonies that are living in a one-sided nest. And that's really important because stable temperature is critical for brood development in these colonies. So what this was telling us is that these two-sided nests have these thermoregulatory benefits for colony growth and thermoregulation, and that there is a benefit to living in a two-sided nest. But you might be thinking to yourself, Michael, don't honeybees usually live in a three-dimensional nest? You showed us those images before of all these multiple combs. Why are you only doing this in an observation hive? And this was actually kind of a nervous thing for me because I was worried that maybe what we were seeing was just all a function of just these observation hives, that we were just kind of barking up a wrong tree. So instead, we just went for it. So what we did was we basically set up these colonies in three-dimensional spaces. So we basically just give them an entire box. And then we take these photographs using a photo rig. This was work that was helped along with Ben Koger, a former cluster person, and Pratibha, a phenomenal grad student in the lab. And because we couldn't really map every single nest individually, we basically came up with a neural network that basically lets us take an image and then classify the different contents to then kind of get a map of what's in that nest. And so I'm only showing you a subset of these nests, but basically, again, what you're looking for is matching colors on opposite sides. This is only 4 of the 6 colonies that we did for this. We've since done it for other colonies as well. And what we basically find is that each comb is symmetrical with itself in the nest, but they are not interchangeable within the same nest or across nests. So what that means is that the bees are creating symmetry at a per comb level, not that they're interchangeable everywhere, okay? So yeah, this front-back symmetry was important even in a three-dimensional nest, and that when we're putting them in an observation hive, we're basically just showing that same comb symmetry, but just now in their entire nest. So at this point, though, I kept thinking to myself, it'd be really cool if we could get all 4 of Tim Bergen's questions, because I feel like we had a good understanding of maybe some kind of like mechanistic idea, developmental stuff. I didn't show you the developmental data, but that we find that it's always symmetrical throughout their nest from initiation onward. So functional benefits, that it's important for thermoregulation and colony growth, but we couldn't get at the evolutionary kind of sense. Where does this, was Apis mellifera some kind of unique, or was it kind of this something that we find across all of Apis? So for this, our question was, well, is it unique to Apis mellifera, or is it shared across Apis? And so again, had to reach out to collaborators, which was awesome to be able to kind of rely on a network. And again, I felt like a horrible person because I was like, Bajari, can you give me some photographs, but I can't tell you why. And she's like, you are a weird guy. And but she sent me these images that she had, 3 different species of Apis. And I'm first just going to show you the images so you can maybe take a look for yourself. Just note that this capping that you see, this is the cap brood. And you might notice symmetry between the 2 sides. And sure enough, yeah, they're very symmetrical. Each nest is symmetrical with itself. They're not symmetrical across. It's really a shared level of symmetry on either side of that comb. So yes, this front-back symmetry exists across Apis, and it's not just unique to Apis mellifera. So at this point, you, and definitely me, was thinking to myself, nobody had noticed this? This is kind of weird. Like you would think honeybees, an organism that people have been working with for quite a long time, a lot of very interesting, capable researchers working on this. And I think it comes down to a couple things. First was that you needed someone dumb enough to do this project in the first place. And by this project, what I mean is just sitting down and saying, I'm going to look at these colonies over time and just trace out the nest contents. This is a heck of a lot of work, and it's not something that people typically do. And I think that was a unique kind of data set. And the second part, to actually sit down and look at the data in their entirety, and actually looking at both sides of the nest next to each other. Because if you didn't look at the 2 sides, then you weren't going to actually notice the symmetry. And this really reminds me of Martin Lindauer and his work, and how he said that the coolest things he ever noticed were the little things that he saw on the side. And so I think that was like a nice kind of callback to German bee science, where the most interesting things you're going to discover in your scientific career are not necessarily the things you're trying to find, they're the other things. But what does this tell us about evolution of three-dimensional nest architecture broadly? If you were to think of some kind of prototypical bee that builds its nest, you could think of like Megaloptogenalis. Maybe they have some kind of material savings by kind of stacking these together, but then maybe there's also some benefit if they're doing some basic thermoregulation. And then you fast forward millions of years, and now you get three-dimensional nest architecture in all these species, and the cavity nesting species, when they moved from these outside places into cavities, they basically pulled that symmetry and they're just kind of like sandwiched onto themselves. So you might be thinking, well, Michael, how important is the parallel comb structure? And for that, I'll have to say, man, leave me alone. I got stuff to do. But we're staying tuned and we're going to do that. And I can't get into that now because it'd be too much. So kind of going a little bigger picture on this and to hopefully connect to other researchers in some ways is maybe you're a multicellular biologist or someone that thinks about multicellular developmental, and there are these molecular pathways like gap genes and bicoid and all these different transcription factors. They create things like symmetry in organisms, and that they lead to things like functional symmetry that is important for things like locomotion in a fly. I think what we're maybe getting at is that this idea that these superorganisms have behavioral pathways that also get us to things like functional symmetry, but are important for different things like homeostasis. And so this would call back to Alan Turing's work, the chemical basis of morphogenesis, but then in a case that we're potentially looking at kind of the behavioral basis for these similar processes. There's a lot of acknowledgments. There's a lot of people that I couldn't even mention, but I definitely mentioned Ian, who was an amazing postdoc advisor and kind of let me do whatever I wanted to, which was amazing. My PhD advisor, some more current collaborators at Cornell, a lot of different other collaborators and lab members, and funding for the NSF and other instances as well. And with that, I'll take any questions.
Thanks, Michael. Amazing talk. Thank you very much. And we're taking questions.
Thanks, Michael.
Hello.
I have a question about the symmetry.
Yeah.
If the temperature is the main driver, I can see how the brood would be symmetrical. But do you think then the stores are symmetrical only because they place it around the brood in a similar way? Or how do you explain the storing?
Yeah, that's a great, so the question of like, okay, the brood's symmetrical because of temperature, then why is your honey symmetrical? And we don't know why the honey is symmetrical. We don't know why the pollen is also symmetrical. The pollen symmetry does get broken if you modify the entrance. So for when we did the one-sided versus two-sided, to control for like entrance area, we only had a one-sided entrance, and that totally breaks the symmetry. You then only have the pollen stored on one side. But one of our most crazy examples of like you're still symmetrical was we had a colony that was on death's door. And we're like, look, instead of having a colony die, let's feed them some sugar water so then they can at least like not die. And they still stored their nectar symmetrical on opposite sides of the comb, even though they were literally about to die. So we still see that they create this symmetry even in their honey stores, but we're not sure why. It might still have something to do with thermoregulation in that if you create honey on both sides, then it blocks it out from brood for both sides. And they also didn't just put it straight up at the top. They actually put it in a band that looked like it was bordering where the brood would be, but that colony had no brood. But it's something that we want to look at more. A classic experiment on that would be to have one-sided combs where we have nectar on one side and brood and see if that impacts the brood's development. Yeah, but yeah, you're totally right, yeah.
Thank you, Michael. Great talk. They build. Do they always only build, or do they also destroy? I mean, do they take off a cell if it's too weird and replace it with a small and a 5 and a 7? How do they manipulate the sort of architecture?
Yeah, so that's a great question. So first up, in the beekeeping folklore, and in a lot of like kind of like published areas, it's like, oh, once they build the comb, it's fixed and it's solid and nothing changes. And as you can expect, because I'm saying this, that's total malarkey. One of our best examples of that is that we were looking at these data sets and we're like, it's weird. The comb shrinks over time, like the overall size of the comb. And so that undergrad, Claire, she actually took that as a side project. And it turns out that what we think they're doing is they actually trim the edges of the comb over time. And we think what they're doing is they're using that comb for cappings, for example, in the wintertime. But it is a continual like shredding of the edges of the comb that they're changing. In terms of the 5/7s, there probably is a lot of mix and match and put up and take down at that initial stage. But once they build those cells and they're kind of solidified, they tend to be fixed. I say tend to be because if you have a 5/7 sided fixture there's probably not a better configuration for it, and it stays there. There are cases though when we take multiple images of like a comb, and you can see like sometimes there's like a faint wall at the base of 2, and they will shred that out or put it back or shred it out and put it back. So I think there's a lot more of editing and re-editing in their nests than we give them credit for.
The builders and the refurbishers, the same cast?
Don't know. Yeah. Don't know. I would love to. Yeah, those are really hard. It's a case of very rare behaviors that you'd have to capture to be able to see those things. So they're difficult ones to get at. But yeah, it'd be a cool. Yeah, I mean, another thing we notice is often in observation hives before winter, you see a lot of these little specks of wax just appear all over the place. And then those tend to disappear over the winter. I think there's a lot more kind of wax deposition happening in nests than we give them credit for, especially considering that as beekeepers, we're so like kind of anal in cleanliness about it, that there's a lot of nest structure that we're not taking into account. Yeah, but yeah, do a PhD with me? No? Oh, bring it on. Yeah.
So when you had your experiment with the heat pads, why don't they grow out of the heat pads with the breeding zone?
Oh yeah, so they do. So on the y-axis, you might have seen there's a time was 10 days. We specifically measured those nests after 10 days because that's the time at which the very first eggs that the queen laid would then have become capped, and then we would be able to say like, okay, this was the initial. Because the bees are, they build a cell and then the queen can lay an egg in it. So that's why we specifically timed it at that point. If you then measured it, 20, 30 days, you might not see as well because then the area of which they have reared eggs is becoming larger and larger. But this was a case where we wanted to use the biology of the bees to be able to show, okay, this is going to give us the clearest indication of their initial cell building and queen egg laying, yeah.
And have you done the opposite and measuring the temperature of the actual hive? Is it not then? So you said the honey is always on top, right? So isn't it?
It's usually at the top, yeah.
So is it not there also because of drying the honey with the higher temperatures, for example? And like when you measure the temperature, do you see like a pattern?
Yeah. Typically, if you take a lot of measurements of colony temperature, typically the brood nest is the warmest and is always kept the warmest. But obviously heat rises, so then you would also have that kind of incidental part as well. I think maybe if I could choose the experiment to do next, that would be like just wouldn't have to actually do the work, but could just get the result. I would love to take 2 colonies and then create a uniform heat profile, and then show that you break the symmetry between the 2 sides, because you've now removed the information from the 2. And I think that would be a good that'd be a good piece of evidence to show that it really is temperature they're using to kind of like a positive negative control kind of thing.
Or constant rotating hive.
Oh yeah, the bees would kill me. Yeah, they'd be so mad. Yeah. But you're right. I mean, whether there are other temperature cues for other nest contents would also be a cool thing. And yeah, it would be something that we need to do.
All right. Thank you so much again, Michael, for the amazing talk, and to you all for coming today. Please be aware, this week on Thursday, we have a guest talk. Andrew King from the UK is here speaking at 2 in the Data Theatre. And next week in the seminar, we have the PhD pitches, so PhD students presenting their work. Feel free to come and don't miss. Thank you so much. Bye.